In this video, we will talk about DNA as a polymer. Polymers are the molecules that are made up of multiple repeating units of smaller molecules known as monomers. You can think of a necklace here. A necklace is made up of multiple beads stringed together, and that will give a long structure. That long structure can be considered as a polymer, and one bead is a monomer. So DNA is also a polymer, which is made up of repeating nucleotide monomers. But the monomer of DNA is not very simple structure. It's a complex chemical structure known as nucleotide. This nucleotide, if you look at this diagram, is made up of three components: a phosphate group, a deoxyribose sugar, and a nitrogenous base. The deoxyribose sugar, the name DNA, deoxyribonucleic acid, comes from this deoxyribose sugar. This deoxyribose sugar has 5 corners: 1, 2, 3, 4, and 5. This is oxygen atom. And there are 4 carbons here, and there is 1 carbon here. So deoxyribose sugar is a pentose sugar. Pentose sugar. This deoxyribose sugar is bonded with nitrogenous bases. There are 4 nitrogenous bases in DNA. And these 4 bases can be grouped into 2 types: purines and pyrimidines. Purines are always double ring structure. So if you look at here, this is a double ring structure. So this is a purine. And purines are A and G. Adenine and guanine are purines. The other two, cytosine and thymine, are smaller single-ringed pyrimidines. So if you look at here, cytosine and thymine. These are single-ringed smaller structure. But guanine and adenine are double-ring bigger structures. So the nitrogenous base can be any of these type, which is attached here to the deoxyribose sugar on the first carbon here. The phosphate group of one nucleotide covalently bonds with sugar molecule of the next nucleotide. So if you look at here, this phosphate group is covalently bonded to the deoxyribose sugar. Now in this deoxyribose sugar, if we go down and see here, this is first nucleotide, this is second nucleotide. And the phosphate group is connecting first nucleotide with the second nucleotide. There is no other connection between first and second nucleotide. And this connection is known as phosphodiester bond. So, as it says here, the phosphate group of one nucleotide bonds covalently with the sugar molecule of the next nucleotide. The sugar phosphate group line up in a backbone. So this is sugar, this is phosphate, this is sugar, this is phosphate. So they make the backbone of the DNA strand, and these are the central ladders. So you look at the backbone here. This backbone is made by sugar and phosphate. Okay, sugar, phosphate, sugar, phosphate. And these colored rungs are nitrogenous bases. The carbon atom of 5-carbon sugar, as I said before, each deoxyribose sugar is a 5-carbon molecule. So this is the first carbon, this is the second carbon, this is the third carbon, this is the fourth carbon, and this is the fifth carbon. We Call these as prime, 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. Okay. The phosphate group is attached to the 5 carbon, 5 prime carbon of one nucleotide and 3 prime carbon of the second nucleotide. So if you look at here, this is phosphate group. It's bonded to the 5 prime carbon of this nucleotide and 3 prime carbon of this nucleotide. Watson and Crick said that DNA is made up of 2 strands, and these strands are twisted around each other to form a right-handed helix called a double helix. These 2 strands are bonded together with the help of hydrogen bonds. These are hydrogen bonds. And you can clearly see that there is a thymine base here and there is adenine base here. In DNA molecule, thymine bonds with adenine only, and guanine bonds with cytosine only. One more thing to be noticed here is the hydrogen bonds between thymine and adenine. There are only 2 hydrogen bonds. But if you look at guanine and cytosine, there are 3 hydrogen bonds between guanine and cytosine. That's how the 2 strands of DNA molecule are held together. So what did we learn here? That in one strand, if you want to bond one nucleotide with the second nucleotide, it's only bonded through phosphodiester bond. But if you want to connect one strand with the second strand, it's bonded through hydrogen bonds between nitrogenous bases. I hope that's clear, and that's the structure of DNA that you need to learn.